Hello everyone, my name is Diksha and I am from the Bodhi Academy. Today I am going to continue with the Biomolecules chapter, which is one of the important chapter from 12th standard chemistry. So in last part we have finished till carbohydrates, reactions, then types of carbohydrates, then their importance, everything. So today we will continue with the second type of biomolecule, which is proteins. Now, as we already know how much proteins are important to us, or what role they play in our in the functioning of our body. So, proteins are most abundant biomolecules in the living system, and chief sources of this protein are milk, pulses, peanuts, fish, meat, and they occur in every part of the body. Now, how the word protein derived? So, it is derived from Greek word proteos, which means primary or of prime importance. So, proteins play a very important role in our body system. So, now let us see uh, how proteins are formed. Okay, so amino acids, they are the basic unit, building block of proteins. So amino acid that contains amino and carboxyl group, that is amino stand for amine group and acid stands for carboxyl group. So they contain both the group. Okay, now all the amino acids, they have trivial names which usually reflect the property of the compound or the source. Now we have different types of amino acids, so we will see one by one here. In this table we have glycine, alanine, valine, then leucine, isoleucine. So these are some important amino acids which are present in the body. Then glycine, it is named since it has a sweet taste, because in Greek glycos means sweet. So glycine word came from the sweet taste. Then these amino acids are generally represented by a 3-letter symbol, and sometimes one letter is also used. So you can see here glycine is represented as GLY, then alanine as ALA, valine as VAL, leucine as LU, and so on. And one letter code is their initial letter. Then classification. Okay, how the amino acids are classified. So they are classified based on their acidic, basic or neutral character, and it depends on number of amino groups and carboxyl groups in the molecule. Equal number of amino and carboxyl group makes it neutral, meaning if you have equal number of carboxyl and amino group, then it is neutral. More amino group, then it is basic, and more carboxyl group, then it will be acidic. Based on this, we have two types: non-essential amino acids and essential amino acids. Now what are these? The amino acids which can be synthesized in the body, they are known as non-essential amino acid. That means we can synthesize them in our body, so no need to take them from outside. That's why they are called as non-essential amino acid. On the other hand, those which cannot be synthesized in the body and they must be taken or obtained through the diet and are known as essential amino acids. So these are not synthesized in the body, but we have to obtain from outside. Then they are colorless, crystalline solids and water soluble, and they behave like salts rather than simple amines or carboxylic acids. Now this behavior is due to their acidic and basic groups in the same molecule. You can see here they form zwitter ion. Now what is this zwitter ion? In aqueous solution, carboxyl group can lose a proton and amino group can accept it. So you can see here in solutions, carboxyl group, it can lose this proton and this amino group can accept that proton, and it forms zwitter ion. Now this is neutral, okay, because it is having both equal and opposite charges but contains positive negative charges. So amino acids in zwitter ionic form, it shows amphoteric behavior as they react with both acids and bases. Then except glycine, all other compounds are optically active. That means they can rotate the plane of polarized light. So glycine is the exception over here. And alpha carbon is asymmetric. This exists in both D and L form, and they have L configurations, and L amino acids are represented by writing the NH2 group on the left hand side. So otherwise in D configuration NH2 group is in the right hand side, but in this it is on the left hand side. Then about structure of proteins, they are connected to each other. Now this is very important, how the proteins are connected to each other through a peptide bond, or it is called as peptide linkage. Okay, in carbohydrates it was phosphodiester, and in this it is peptide linkage. In carbohydrate it was glycosidic linkage, and in proteins it is peptide linkage. Then reaction between the two molecules or of similar or different amino acids proceeds through the combination of the amino group of one molecule and carboxyl group of other molecule. So you can see here they are combined together. These two amino acids are treated together, and this is a peptide linkage which is formed from carboxyl group of one molecule and amino group of other molecule, and this is what called as a peptide linkage. Then the product is called as dipeptide because we are combining two amino acids, and when carboxyl group combines with the amino group we get dipeptide, that is glycylalanine. If third amino acid combines to dipeptide, it is called as tripeptide, and so on. And if you have more than 10, then it is called as a polypeptide. And then we will see on the basis of their molecular shape, proteins are classified into two types: fibrous and globular proteins. Now what are fibrous proteins? In fibrous protein, the polypeptide chains run parallel and they are held together by hydrogen and disulfide bonds, and fiber like structures are formed. So protein, they are generally insoluble in water, and fibrous protein examples are keratin and myosin. Keratin is basically present in hair, wool and silk, and myosin is present in the muscles. So these points about fibrous proteins are very important and they are asked in the exam along with the examples. Then about globular proteins, here they don't run parallel but they coil around to give a spherical shape. So, and these are soluble in water, and examples are insulin and albumins. Then structure and shape of proteins can be studied at 4 different levels, that is primary, secondary, tertiary and quaternary. Talking about primary, they have one or more polypeptide chains, and in a protein has amino acid linked with each other in a specific sequence. So any change in primary structure, the sequence of amino acid creates different protein. Then about secondary, in secondary we have two types: alpha helix structure and beta pleated structure. So in secondary type, alpha helix is one of the common ways in which the polypeptide is forms all possible hydrogen bonds, and beta pleated structure is all peptide bonds are stretched out to nearly similar maximum extension, and the laid side by are held together intermolecular hydrogen bonds. So here what you need to understand is the types, two different types, and how they are bonded to each other. Then tertiary structure, it represents the overall folding of the polypeptide chain. So further folding of secondary peptide results in tertiary peptides. The forces which stabilize secondary and tertiary structure are hydrogen bonds, disulfide linkage, van der Waals, and electrostatic forces of attraction. So these are the forces which stabilize the secondary and tertiary structure. Then last is quaternary structure. So quaternary structure means here the spatial arrangement of the units with respect to each other is called as quaternary structure, and here you can see all the four. Primary means it's same straight line linear kind of structure. The secondary is meaning linear structure is further coiled. Then when you coiled it more, it is tertiary structure, and the complex coiling will give result to quaternary structure. Now very important concept here is denaturation of proteins. See, when we boil egg, what happens? It becomes harder. So protein found in the biological system, it is having a 3D structure, and biological activity is called as a native protein. But when it is in native form, it is when it is subjected like a physical change, like change in pH, change in temperature, then their hydrogen bonds are disturbed, and due to this they unfold and helix get uncoiled and protein loses its biological activity, and that is what called as denaturation of proteins. So this is denaturation of proteins. Now in simple words you can say that when proteins are subjected to change in temperature or change in pH, they lose their biological activity, and that is called as a denaturation of protein. And during denaturation, secondary and tertiary structures are destroyed, but primary structure remains intact. An example here is, common example is coagulation of egg white on boiling. Then one more example is curdling of milk. Then next we will see about enzymes. As we have learnt in surface chemistry, enzymes are the biological catalyst. So life is possible due to coordination of various chemical reactions in the living organism, and to carry out those reactions more efficiently we require enzymes. So this process involves sequence of reactions, and all the reactions in the body under very mild conditions. So this occurs with the help of certain biocatalysts called as enzymes. Now here one example we can see enzyme maltose, which is having C12H22O11 formula. When it is converted to glucose, we use maltase enzyme here. Okay, so maltase is used as an enzyme to convert maltose to glucose. Then sometimes enzymes are also named after the reaction where they are used. So mostly they are named after the reaction where they are used. Then mechanism of enzyme action. Enzymes are needed in very small quantities for the progress of a reaction. So they are said to reduce the magnitude of activation energy. That means if hydrolysis of sucrose requires 6.22 kilojoules per mole energy, and activation energy is only 2.515 kilojoules per mole when enzyme is used. So this is how we can reduce the energy. Next is vitamins, which are very important biomolecules for our body. Now vitamins, they are required in very small amounts, but their deficiency causes specific disease. So they are very important for our body. So most of the vitamins, they cannot be synthesized in our body, and plants can synthesize almost all of them. So different vitamins belong to different chemical classes and generally regarded as organic compounds required in diet. Now this is a specific definition for vitamins. Organic compounds required in the diet in small amounts to perform the specific biological functions for normal maintenance of optimum growth and health of the organism. They are designated by alphabets A, B, C, D, etc. and they are further named as subgroups, that is B1, B2, B6, B12. So these are the subgroups. Excess of vitamins is also harmful. That's why only in limited amount we should consume the vitamins in order to continue the growth. Now classification of vitamins. There are two types of vitamins, fat soluble and water soluble. Fat soluble means which are soluble in fat and oils, but they are insoluble in water. Now this includes vitamin A, D, E, K, and they are stored in liver and adipose tissues. Under water soluble vitamins, we have two groups, B group and vitamin C. They are water soluble and they are grouped together. So they must be supplied regularly in the diet because they are readily excreted in the urine. So here we can see what are the sources and what are the deficiency diseases caused when the vitamins are not supplied regularly. So vitamin A, we can get the vitamin A in fish, liver oil, then carrots, butter and milk. And its deficiency can cause xerophthalmia, which is a hardening of cornea of the eye. Vitamin B1, which is also called as thiamine, it is present in yeast, milk, green vegetables and cereals, and deficiency of this can cause beriberi and retarded growth. Vitamin B2, which is called as riboflavin, we will get it in milk, egg white, liver and kidney. And chelosis is the disease which is caused when you have deficiency of vitamin B2, and also digestive disorders and burning sensation of the skin. Vitamin B6, called as pyridoxine, is found in yeast, milk, egg yolk, cereals and grams, and it causes convulsions. B12 is found in meat, fish, egg and curd, and it causes pernicious anemia, that is deficient RBC deficient in hemoglobin. Vitamin C, also known as ascorbic acid, it is found in citrus fruits, amla and green leafy vegetables, and its deficiency causes scurvy, that is bleeding gums. Then vitamin D we already know, get from sunlight, fish and egg yolk, and its deficiency causes rickets, that is bone deformities, and osteomalacia, that is soft bones and joint pain. Vitamin E is present in vegetable oils like wheat germ oil, sunflower oil, and increased fragility of RBCs and muscular weakness. The disease caused due to the deficiency. And last is vitamin K, which causes increased blood clotting time if it is deficient, and is present in green leafy vegetables. Now next type of biomolecules is nucleic acid. In nucleic acid are very important, plays a very important role. So nucleic acid, it is basically present in the DNA. So the particles in the nucleus of this cell responsible for heredity, they are called as chromosomes. This we have learned in biology, and which are made up of proteins and another type of molecules, they are called as nucleic acids. So they are two types, deoxyribo and ribonucleic acid, so DNA and RNA. So nucleic acid are long chain polymers of nucleotides. Now this line is very important. Nucleic acids are long chain polymers of nucleotides. Now what are the chemical composition of nucleic acids? So complete hydrolysis of DNA or RNA, they yield pentose sugar, phosphoric acid and nitrogen containing heterocyclic compound called as bases. So these are the products we get when we do the hydrolysis of DNA and RNA. In DNA, sugar moiety is beta D2 deoxyribose, whereas RNA it is beta D ribose. So this is a difference in that. Then DNA contains 4 bases, that is adenine, guanine, cytosine and thymine, and RNA also contains same bases, only the thymine base is replaced by uracil. So it's not present in the DNA. And this is structure of nucleic acids. A unit formed by the attachment of base to first position of the sugar is called as a nucleoside. So here we can see nucleotides. Now here linkage is different. Nucleotides, they are joined together by phosphodiester linkage between 5' and 3' carbon of the pentose sugar. So this linkage between them is called as a phosphodiester linkage. Okay, then in secondary structure of RNA, single stranded helix is present, which sometimes holds back on itself, and RNA are of 3 types. They perform different functions. So we have 3 types of RNA, that is messenger RNA, ribosomal RNA and transfer RNA, which are called as mRNA, rRNA and tRNA. Then about DNA fingerprinting, it is known that every individual has their unique fingerprint. So these occur at the tips of the fingers and have been used for identification for a long time, and this can be altered by surgery. So now this can be used, applications of fingerprinting is in forensic laboratories for identification of criminals, then determine paternity of an individual, then to identify the dead bodies in any accident by comparing DNAs of parents or children, and to identify racial groups to rewrite the biological evolution. Then biological functions of nucleic acid. DNA is a chemical basis of heredity and may be regarded as a reserve of genetic information. It is exclusively responsible for maintaining the identity of different species of organism over millions of years. This is how DNA is very important to us. Then next is hormones. Hormones, they are the intercellular messengers, and they are produced by endocrine glands in the body and are poured directly in the blood stream, which transfers them to site of the action. Some common hormones that we came across in our previous classes, that is estrogen, testosterone, insulin, thyroxine, So these are the hormones which play very important role in our body. Then insulin, it helps to maintain the blood sugar level in the body. Then thyroxine, to reduce the thyroid gland, it is a iodinated derivative of amino acid tyrosine. Okay, so yeah, this is about hormones, and this we already studied in the biology chapter. So from this chapter, the important part that we have to focus are evidences of glucose structure, then structure of the glucose, then about disaccharides, that is fructose, maltose, sucrose, maltose, lactose, and then structure of fructose, about vitamins, proteins, amino acids. All these are important along with the nucleic acid. So with this we finish the biomolecules chapter, and I will come with the next chapter in the next video. Thank you.